Now it should be good to go. Finally.
Yeah, perfect.
Yeah, finally. Okay. Sorry, guys. Technical issues. You notice you're starting to get into the professor range when you start to messing up with the slides. So it's common. Getting back to it, I did my bachelor at Unifesp in São José dos Campos. I had a short stay in the psychology course from the Federal of Minas, but then I switched back to biomedical engineering, where I did my master's, again at Unifesp. And I'm now doing my PhD at Neuroscience and Biomedical Engineering here at Aalborg University. So what is TMS-EG? We're basically working with a combination of two techniques, transcranial magnetic stimulation and electroencephalography. The second one, the EEG, is more common. I believe that most of you have already at least heard of it. It's quite common to be used in some hospital settings. And TMS, it's not as common, but it's growing and it's being used for some conditions. And it's basically a way of non-invasively, you can stimulate the brain with the TMS, and you can monitor the brain oscillations with the EEG. We can use that to probe different brain areas and networks, and you can study clinical populations and basic neurophysiology using the combination of these tools. That is the main purpose of this method. So, mentioning the basic principles of TMS. TMS is basically a coil, usually two coils packed together, where you generate a brief time-varying magnetic field. So if you already had the sad opportunity of joining the physics course, you might remember that when you vary a magnetic or a current inside a coil, you will produce a magnetic field. So we do that very shortly and very intensely. So we create a strong magnetic field that will induce an electrical current inside the brain, leading to neuron depolarization. This method is used in hospitals to assess cortical excitability, connectivity, and plasticity. And one of the examples that where you can use it is to evoke motor potentials. So you can assess cortical motor responses. The way you do that is that you can stimulate the region of the primary motor cortex, for instance, and you will see a hand movement if you manage to stimulate the right area. And you can check the speed of this, the signal propagation, the intensity of the amplitude of the movement, and you can check, for instance, if the tract is functioning well, or if it has any defect in the conduction of the signal. Also, please feel free to interrupt me if you feel like you have any question. Sometimes it's better to bring it on time so you don't forget later. So please feel free to interrupt me. Regarding EEG, it's a quite simple tool. You're basically measuring electric activity that's generated by postsynaptic potentials in cortical neurons, mostly pyramidal neurons, because they are aligned in a way that they stand up perpendicular to the scalp. So the differences in current that they produce are usually larger. So you have these groups of pyramidal cells that will fire up together, and you capture the signal with the EEG, which are basically electrodes positions over the scalp. This tool has some limitations due to volume conduction, but still it's quite useful. There are many uses in the clinic. Some of them, for instance, you can use it for brain-computer interfaces. It's a quite hot topic nowadays within neuroscience. One example is steady-state visual evoked potentials, where you can have stimuli presented at different frequencies, and then on the occipital cortex, you can capture what is the brain attending to. So you can, for instance, have 3 buttons, each one vibrating at a different frequency, and you can know somewhat where is the attention span of that person. It's very interesting for rehabilitation and brain-computer interfaces in general. In the hospitals, it's used for seizure detection when you have a patient that needs to be monitored. And also, very importantly, the bispectral index is one of the tools that they use to assess depth of anesthesia. So you can monitor patients under anesthesia. And it's also EEG based. But I would like to give a little bit more attention to the event-related potentials. In this time, I have an example here, the mismatch negativity, which is the type of evoked potential you have when you are presented with a stimuli that is non-expected. So in this case, this figure here, we have on the top a stimuli that was presented repeatedly, and on the bottom, a stimuli that is different than the previous one, an auditory in this case, that would appear randomly within the sessions, and they have different properties. I just wanted to bring this because sometimes it's hard to visualize. So you have the EEG, and then you present some type of stimuli or cognitive task multiple times across the session. Usually, the stimuli will not have a big amplitude when compared to the EEG in general. That's why you present it a lot of times. And then when you average across these stimuli, you can have evoked potential, as we call it, which is basically the average of the evoked. So you can see that on the baseline of before zero, we have something that is close to zero because everything that is random tends to cancel within the trials. And what is common to all trials is preserved on the average. And that's why we have a peak after zero, which is the evoked potential. And now it's quite clear, right? Why would you like to combine two, or how can you do that? So basically, you have a tool that allows you to excite different brain regions, and you have another tool that allows to capture evoked potentials and cortical oscillations in general. So why not combine the two of them? So with that, you have the opportunity to look at local responses close to where it's stimulated, and also distant neural network activity, because you know where you're entering the system with the coil, and you can measure the entire brain response. This is currently the unique non-invasive assessment of effective connectivity that we can have in humans. The main advantages of using this tool is its high temporal resolution. When you look at fMRI, for instance, you're in the scale of 2 seconds, maybe 1 second if you have a very expensive machine. We can probe different regions, as I mentioned, so you can go for motor or non-motor areas. If you move prefrontal, you might be engaging in cognitive aspects of cognition, brain function. And it's, as I mentioned, it's non-invasive and safe for most populations. Some of the cases where we usually don't use TMS are basically people parent-child and epileptic patients, although it's not fully contraindicated for the epileptic ones. And if you have metal implants on your head, such as the deep brain stimulation or residual metal from some type of fracture or surgery. The main disadvantages of this technique is because it's quite hard to use. It has a lot of technical challenges regarding the pulse. You can imagine you have something that is measuring electrical activity, and then you come and hit it with a very large, intense magnetic field. You have also artifacts that are common to all types of EEG, so such as eye movement and muscle artifacts, mostly from the temporalis muscle and the neck muscle. The coil positioning also is not quite the easiest task. You need to have a neuronavigation or something that can allow you to keep track of where you are with the coil regarding the position with the brain. And also, there's a lot of analysis challenges because you don't have a standard way of pre-processing and processing this data, and it's highly variable across subjects. Here on the right, we have an example of a TMS evoked potential. And basically, what you can see on this figure there, we have 165 and 56 pulses. Again, the baseline is close to 0 because we took the average of this number of trials. And we have this complex brain response after 0 milliseconds. And you can see here what I mentioned about high temporal resolution. We are working on the scale of milliseconds here. It's quite fast. Still, it's quite informative. What do we need to work with this? So we need a EG cap with some electrodes and an amplifier. We need to find where we want to position the coil. So usually we do that with the help of a neuronavigation. And one example I can give is the primary motor cortex. You can do that with the patient or the participant fMRI, or since we have this tool, which I think is quite interesting, you can go functionally to the motor cortex. What I mean by that is that since you can stimulate and get a response out of the hand, because you can find a hand, you stimulate and the hand moves, you can find where you have the highest amplitude of movement. You adjust the coil there, you find the hotspot, as we call it, which is the brain area where the hand representation is. And then you slowly reduce the intensity until you have no movement, because you want to avoid sensory feedback. Because I think the interesting part here to take out of this is basically you're measuring brain. So we need to be careful not to produce anything other than what we want to. One instance of that is the noise mask. So participants, they need to be using earphones because they can't hear the coil click. Otherwise, alongside with the pulse that you're delivering to the cortex, you're also producing a click sound with the machine, and the click itself will produce a brain response, an auditory evoked response. So we need to avoid that also. We usually give around 100 and 200 pulses, and we do a lot of artifact removal methods. So we do the artifact interpolation, we reject bad channels and bad epochs, bad trials. We usually use something like an ICA, then we filter, we do the bad channels interpolation, and we get up to this signal here, which is the TAP. I will call it TAP from now just to make it shorter, but it's basically the TMS evoked potential. Some of the outcomes that we can use from this signal here, we can look at the peaks, that is the amplitude of the response, and that itself reflects excitation and inhibition of certain regions of the brain, depending on where it's stimulating. And it's believed that it is associated with GABAergic neurotransmissions. And you can also look at the frequency content of this response using methods such as the event-related spectral perturbation, intracerebral coherence. You can extract the natural frequency of the system, and you can look at the effective connectivity. So you can look at how the signal is propagating across different brain regions. I don't want this to feel too much abstract. So a way to think of this is basically you have a tool that allows you to knock on a certain place in the brain. What we are doing here, we are knocking and recording with a microphone. So you can have information about how that region oscillates. And you can already have the intuition with that knocking and the microphone. Let's say you have something like, I don't know, this mug, ceramic, and you have a piece of wood. So you hit the ceramic and you hit the wood, they're different. The idea here is that if you have an pathology or a neurological condition where you know that a certain region is implicated, you can go there on healthy participants and you knock there, record it, and then you go on the pathophysiology or the clinical population, and then you knock there and you can see if there's any difference. You can try to have a better understanding of the mechanisms that might be involved in certain neurological conditions. Let's just drink some water. So, as I mentioned, this tool has been widely used to understand neuromechanisms in physiology, although it's a quite recent developed. It has around 18, 20 years that it's been really like worked on. Although TMS is older, TMS-EG was just recently put into practice because we didn't have electrodes that could be used alongside with the TMS. Now we have. And then we have some studies trying to describe a little bit better different brain regions and characteristics and how they communicate using this tool. But I think the most important part, at least for me and maybe for many of the ones that are here that are from the biomedical engineering course, neurological disorders and clinical conditions, such as disturbance of consciousness, schizophrenia, and depression, which are just three of the examples that I was planning to bring. So when we look at disorders of consciousness, I decided to take this work. It's from my supervisor there in Brazil when he was in Italy in 2013. They showed that with this technique, you could differentiate between healthy participants that are awake and that are under effect of anesthetics using a complex method. It's called a perturbational complexity index. But in essence, what it is capturing is how complex is the response. So how much does it change across regions? Is it the same across the entire brain? What is happening with communication terms? How does it change? And more importantly, and I think here is where things get very interesting in this case, is that they followed a couple of patients that were in coma. They were under they were in the coma. Some of them due to lesions, and they followed these patients from coma to awake, and they showed that this index it increases when they return to conscious, and when they not, it usually stays low. So in some way, they were capturing with this the level of unconsciousness of people, and it's been a couple of years since then. Now they are currently trying to move this into the ICU in the United States. There's a group in Boston, if I remember well, that is working with it. So you can have a prediction if patients will come out of coma. And you can imagine how much this is impactful for families and how this could help resource management and also how many lives this could impact in that sense. Another very interesting study now regarding schizophrenia is the study from Donati. This group from Pittsburgh, they have a couple of studies. This is not the only one, where they investigate with TMS-EG, they investigate schizophrenia. I think this one is a very interesting one because they work with first episode patients. So you have usually teenagers where they have their first maniac episode or the first paranoid episode. They have the first outbreak. So it's not someone that is schizophrenic, but it's someone that they call schizophrenia pre-domus. So they have the first episode, and then they go to the doctor. They do the TMS there at Pittsburgh, and they showed that you can have an identification early, on the very early stages of schizophrenia, that there is an alteration in the circuit of the prefrontal cortex. So here again, this is a tool that can be very helpful in the clinics. It's still not there. We're still far from that. But you can understand a little bit better the mechanism of the pathology, and you can also predict the progression of it. I was planning on bringing this one from major depressive disorder, but I just wanted to point out that there's a lot of pathologies you can investigate. So, and also you can start into this challenging field because many of disorders that we have, many of the neuro disorders that we have, they share some of the aspects of the others. So, for instance, we know that bipolar disorder, major depression, and schizophrenia, they affect somewhat the prefrontal cortex. So when you look at that, it might be not much specific towards the disease, and there is where the work is now. So we're trying to investigate better how could we specifically find decent biomarkers for different disorders. So we lack artifact removal techniques that can be automated. That would be super helpful for the community. Standardized acquisition protocols, pre-processing, analysis. Also, software is something that we lack. Most of the signal processing and analysis now is done usually by a programmer or doctors who know a little bit how to work with MATLAB. They're still slowly moving to Python. Yes, we can gain some insight in neurophysiological mechanisms. The idea here, trying to find these biomarkers in different clinical populations and many other uses. Yeah, I would like to just point out that neuroimaging techniques, they have enormous potential to assist research in clinical practice. TMS is just one of these tools. I'm not crazy about it. I think it's super cool, but all type of neuroimaging can provide different information about the brain and clinical conditions. And also the changes in this cortical connectivity, they might be associated with specific neurological conditions, and there's a lot of space for us to help improve diagnosis and intervention in these pathologies. Thank you for your attention, and I will open for questions, discussion, and I have an extra material if you guys would like to, after we talk a bit